eastern oyster is one of the most iconic species in the Chesapeake Bay. For more than a century, oysters have made up one of the region's most valuable commercial fisheries, and the filter feeder continues to clean our waters and offer food and habitat to other animals. But overharvesting, disease, and habitat loss have led to a severe drop in oyster populations. Chesapeake Beach Oyster Cultivation Society is working to manage harvests, establish sanctuaries, overcome the effects of disease, and restore reefs with hatchery-raised seed, an effort to bring back the oyster. Before the discovery of America and the rise of industrial oyster operations, oysters were abundant in the bay. Archaeological evidence shows local Native Americans began eating the delicious oyster over 5,000 years ago. When colonists arrived to the New World, the Chesapeake Bay was much clearer than it was now, partly because of the cleaning powers of the oyster. In fact, there were so many of these filter feeders that the name Chesapeake is thought to be derived from a Native American language, meaning Great Shellfish Bay, and visibility would sometimes reach 20 feet. Human impact on oysters were evident then too. One archaeological site measured oyster sizes near Maryland's old capital, St. Mary City, from 1640 to 1710. In 1640, when the city was still small, oysters measured 80 millimeters, and in the city's maximum population, in 1690, they measured to 44 millimeters. When the capital moved to Annapolis, the population moved with it, and by 1710, the oysters were back up to 80 millimeters. During the Industrial Revolution, several new technologies were introduced to the Chesapeake area, which allowed more intensive oyster harvesting. The first was the invention of canning, which allowed oysters to be preserved much longer and create a demand for oysters across the world. A second invention was dredging, which enabled oyster harvesters to reach untouched depths of the Chesapeake and clear entire oyster beds, where oyster carcasses would be grounded up and used as filler in roads and a source of lime and fertilizer. The final invention was the introduction of steam-powered ships and railroads, which made the distribution of oysters easier, enabling merchants to sell oysters almost anywhere. By the 1880s, 20 million bushels were being harvested from the bay each year. By the 1920s, harvesting would be down to just 3 to 5 million bushels per year because of overharvesting. Overharvesting is why the oyster population has been depleted to just 1% of its historical level. Oysters were being taken much faster than they could reproduce. Also, many of the shells which were vital to the ecosystem were being taken and not replaced, destroying the aquatic ecosystem. By the 1920s, oyster populations stabilized for a time by returning oyster shells back to the bay. But in the 1950s, oysters had to deal with two diseases, called Dermo and MSX, which decimated the remaining oyster population. The two diseases were unheard of, and in the Chesapeake, and were thought to be brought from Asian oysters. The rise of populations around waters also contributed to the loss of oysters. Runoff from construction, pollution, and destruction of SAV all damage the water quality in which oysters need to thrive. From overharvesting, disease, and pollution, the current oyster harvest average is less than 200 bushels per year. The decline of oyster is very bad for the ecosystem of the Chesapeake Bay because oysters are so important. Oysters are considered a keystone organism, and without the presence of the oyster reef, other critters would not be able to move in and colonize the reef. The most widely known ecological function of the oyster is that they filter the water, filtering almost 50 gallons per day. Oysters are considered the vacuum cleaners of the Chesapeake Bay. They filter the water, removing organic and inorganic particles from the water column, resulting in cleaner water which positively impacts other species. Oysters can selectively choose and feed on microscopic plankton and algae, removing the algal biomass from the water. Oysters also remove other suspended solids from the water column and package them into bundles, which is then utilized by other organisms on the oyster reef for food. The presence of oyster shells creates a hard bottom substrate that provides habitat for other bay organisms. Many critters depend on the existence of the shell to grow. Barnacles, mussels, and anemones all require a hard bottom on which to attach and to grow. Oyster reefs also provide shelter and spawning areas for the bay residents. The gapping oyster shell provides a substrate for the eggs to be attached to, while offering protection from predators at the same time. The nooks and crannies of the reef formation offer habitat to different species of worms, fish, and mud crabs. The presence of these organisms attract larger predators, which in turn attracts even larger predators. The existence of an oyster reef truly creates a dynamic environment. These many functions of the oyster is why they are so important to the bay. And that is why an organization like Seabox works so hard to restore the oyster population. But to do so, you need to understand the life cycle of the oysters. After oysters are harvested and eaten, Shell Recycling Alliance partners, which include 225 seafood services, save the oyster shells, which then are taken to oyster hatcheries where they are aged and washed. Then scientists introduce oyster larvae, which attach to the recycled shell. These young oysters are now set to grow in floats that hold bags of fertile oyster near the surface. In these cages, they are protected from predators, where they fully grow without problems of overcrowding or competition for food. Once fully matured, the oysters are placed back into the bay in specific locations, where they will be untouched and allowed to further grow. In order to properly grow oysters in floats, the water conditions have to be ideal for oyster growth. Seabox is continuously testing the water of the local fishing creek river to make sure it stays at an optimal level for oyster growth. They also show local elementary schools the importance of oysters and how to test the water during field trips to their oyster aquarium. During these field trips, students are taught to measure turbidity, dissolved oxygen, pH level, salinity, temperature, and speed of current, all important factors in oyster growth. The first characteristic of the river that students measure is the speed of the current, which is important because the current can't be too strong, or the oysters will be washed away, and the current can't be too slow or stagnant, or parasites that cause disease will thrive. Students measure the speed of the current with a current meter, a long rod with a propeller on the end. A wire that climbs the pole leads to an electric meter that reads the current speed. Students place the propeller end into the water, and the meter shows the speed of the current. The speed measured 0.05 feet per second, which is not even 1 mile per hour, perfect for oyster growth. To test the other characteristics of Fishing Creek, students collect the water directly from the river and place it into red buckets, each being used to test a different characteristic. The students use the first bucket to test dissolved oxygen, which an oyster needs a certain amount to breathe. Too little dissolved oxygen can leave oysters vulnerable to the disease dermo. Students use a dissolved oxygen sensor, which is connected to an electrical meter that tells the dissolved oxygen level. Students swish the sensor around in the water, and the meter tells them the level of dissolved oxygen. The meter reads 0.6 milligrams per liter, which is above the required 0.3 needed. The next bucket is used to measure salinity, the measurement of salt in the water, which greatly influences growth and the resistance to disease of an oyster. Students use a salinity meter, much like the dissolved oxygen meter. They swish the sensor, and the meter tells them the level of the salinity. The meter reads 0.6%, which again is above the required 0.4%. The last bucket is used to measure the pH level. A too high or too low pH can kill young oysters. It's important for the pH to remain within 6.5 and 8.8. Like the other meters, the pH meter has a sensor, an electric meter that tells the pH level. This meter also tells temperature. Students swish around the sensor, and the meter tells the pH level and temperature. The meter reads 7.9 and a temperature of 68.5 degrees, falling into the pH range and the temperature range of 38 to 86 degrees. Lastly, students test turbidity, the cloudiness of the water. High turbidity inhibits light from reaching lower levels of water, which can inhibit the growth of SAV, which in turn affects the species of fish and shellfish. Students use a Secchi disc, a disc with 2 quadrants of black and 2 of white, to measure turbidity. The disc is connected to a robe with red lines that each represent 6 inches. Students lower the disc until they can't see it and record the amount of inches it went down. Students recorded 24 inches, which is above the required 20 inches. 2 other characteristics of the water that they sometimes will test is phosphate and nitrate. Many times in aquatic ecosystems near construction and other civilizations, nitrate and phosphate levels are high and can potentially cause the death of fish and shellfish. Luckily, for the Fishing Creek ecosystem, the river has a natural marsh that thrives off the phosphate and nitrate, using the 2 substances as a fertilizer. The marsh protects the river from too high of levels, and in result, protects the aquatic life. Because of the efforts of Seabox and the citizens of Chesapeake, and with the help from the marsh, Fishing Creek is perfect for growing new oysters and restoring the population to the bay.